Hello here. In this video we will discuss about the endocytosis, so the phagocytosis and pinocytosis. So overall we will just understand about the phagocytosis. So the phagocytosis is the numerous cells are able to ingest the foreign materials but ability to increase activity in response to opsonization by antibody in the immune system and complement system. Basically, is the it is so it requires antigen specificity. So the antigen specificity requirement for the engulfing of the content, antigen content. So the monocyte will proliferate to macrophage, and it is also able to phagocytose and neutrophils, antigen representing cells also. But here if we will understand about the neutrophils and macrophage. So we will understand about the neutralization as well as other things which that is content will be unable to infect in the cell. We will discuss how it's processed. So steps of phagocytosis and intracellular destruction of microbes. Just we will understand in this video. So the phagocytosis just. So the chemotaxis is the first sign which that is cell produce interleukin as well as chemokines. These chemokines will produce where the production of the chemokines. So the cell will follow toward the antigen is known as the positive chemotactic movement of the cell. And in this way the attachment will be occur with the antigen with a receptor. This receptor should be specific for the endocytosis. After all, the endocytosis will be occur in this way. The endocytosis, the vacuole formation. After the vacuole formation, the lysosome will be bind and the killing through hydrolytic enzyme as well as other cationic proteases, proteins and ascorbate, elastase and myeloperoxidase and lactoferrin. So the this all thing is used for the killing. After the killing, that is digestion. And digestion to show the antigen representation for showing that through MSC class 2 and 1, it means the major histocompatible complex will show. And let's begin to understand with the macrophage with a bigger picture for understanding the best and good manner. So let's begin to understand from here. So first of all, I want to say that first of all, I will draw the cytoplasm and cell membrane. So cell membrane is made up of bilayer of phospholipid, which that is extracellular matrix are present in the outside of the cell, while inside is the intracellular fluid. So ICM and ECM, and in this way here is the fluid will move inside. So in this way this is the fluid, and it means this is known as a pinocytosis, a pinocytosis. And after this fluid will move inside, this fluid can be toxin or other things will be neutralized through lysosome binding. So after the binding with the lysosome contain hydrolytic enzyme for neutralization of the pathogen or pathogen associated molecules or liquids. But in the case of particles, for example, here is known as phagocytosis because solid particle will engulf and pattern recognition receptor here which that recognize the pattern of the antigen, and after this recognition that will be cascade of phosphorylation will engulf this bacteria or pathogen. And in this way, on the other hand, the C3 receptor which contains C3 protein with the complement system to engulf, while here is the Fc receptor which that is used for the Fc portion of the antibodies after the opsonization of the antigen through a plasma cell and B memory cell. And this plasma cell will opsonize through antibodies and opsonin protein in the case of complement system which that is used for the movement inside through Fc receptor or C3 receptor. So let's begin to understand here is the chemotaxis first. This chemotaxis means the following. The cell will follow toward the chemical. It is known as the positive chemotactic movement. So the inflammatory mediators produced from infection site to recruit the macrophage and monocyte as well as neutrophil and other cells, mast cells, natural killer cells, etc. So this is the attachment occurs by the opsonized antigen associated molecular pattern. So after this, the molecular pattern you can see and understand as well. So after this, the endocytosis will be occur through a pathogen, so is known as the phagosome. This phagosome, why? Because it is solid, and in this way it will move inside is the phagolysosome formation. So the phagolysosome means because the phago means the eater, eated, and in this way the lysosome will be bind. A lysosome will come from after the maturation with the lysozyme enzyme, hydrolytic enzyme, and other through Golgi bodies and endoplasmic reticulum for maturation through a transcription factor to translation to produce lysosome. Lysosome will bind with the phagosome to phagolysosome formation. And the autophagosome is also produced, which that is also bind with the phagolysosome for increasing the or efficiency of the enzyme. For example, lysozyme and cationic proteins, ascorbate plus copper, elastase, and myeloperoxidase with chlorine and the lactoferrin, which that is all thing is used for the neutralizing the antigen or bacteria or virus. So the arginine is also produced, which that is arginine will produce the nitric oxide. This nitric oxide and other content which that enzyme will neutralize. While in the case of, on the other hand, one more important thing is that it is the aerobic process. In this way, the oxygen will move inside from extracellular to the intracellular. So the endocytosis will occur, vacuole formation, and after the vacuole formation, digestion to antigen representation will be occur. So how antigen will produce, how antigen represent, so we will also understand. So let's begin to understand. Here is the oxygen which that is moving inside, and reactive oxygen species formation due to the superoxide dismutase enzyme. And the superoxide, after the accepting of the electron, it will use for the in the presence of superoxide dismutase to convert the in the presence of NADPH oxidase enzyme to produce the hydrogen peroxide. And in this way, the hydrogen peroxide will separate the water is the digestive enzyme is also water, and the water as well as the hydroxyl group, hydroxyl free radical, and oxygen will be separate also. This hydroxyl free radical is used for the damaging of the wall of the pathogen, so antigen. So at the digestive enzyme will produce this type. It is the inactive antigen formation. After this inactive formation, here is the digestive enzyme can be acid phosphatase, lipase, used for the digestion of lipids, and phosphate group, and elastase, and beta glucuronidase, and cathepsin. This all enzyme is used for the digestion of the pathogen. After this digestion, it gives us a one part of the antigen for with showing with the MSC class 2 molecule, major histocompatible 2. And in this way, the antigen representation through T cell, the T helper cell, and after this T helper cell with the CD4 molecule bind with the MSC class 2 molecules, etc. And in this way, here is the T cell will be activate and activate to the CD8 for the cytotoxic T cell to activate to kill this cell. On the other hand, it will also trigger the adaptive immunity to produce more antibodies for combating the microorganism growth. So all these things about the phagocytosis. Thanks for watching. Please make sure to subscribe, like, and share. Ask the question for answer and comment about the artwork. You can give me feedback.